My name is Kaci Jinright, and this is my video on DNA replication, transcription, and translation. First, we'll start with DNA replication, which is pictured here. DNA replication is the process of producing two identical replicas from one original DNA molecule. We start with one double-stranded DNA molecule, which you can kind of see right here. The initiator protein will bind to the origin of replication, which leads to unwinding. The helicase, which is right here, will also bind to the lagging strand. Helicase breaks the hydrogen bonds between the complementary nucleotides, which separates the once double-stranded DNA into a leading strand and a lagging strand. The initiation point, where the splitting of the two strands occurs, is called the origin of replication, or the replication fork, which you can see right here. So let's talk about replication of the leading strand first, which is the top one up here. RNA primase will create a primer, which is these little red things down here. It's not up here. They didn't picture it up here, but just imagine it. It would be right here. Made of RNA nucleotides, which binds to the 3' end of the template strand. This initiates replication. Replication is continuous in the leading strand. See, there's no breaks in it up here. DNA polymerase 3, which is this thing right here, will encode the new strand, which is the leading strand. Here. Now for the lagging strand, which is the bottom strand. Replication is not continuous in the lagging strand. RNA primase creates a primer made of RNA nucleotides, just like in the leading strand, which will bind to many different places. So there's 4 pictured here on the template strand. DNA polymerase 3, again, will fill the gaps between the RNA primers with complementary nucleotides. These gaps are called Okazaki fragments. To the fragments. The DNA polymerase 1 will remove the RNA primers, the red things. Here's DNA polymerase 1. And they will provide complementary nucleotides to those regions. DNA ligase will come along and ensure that all the Okazaki fragments are joined into a continuous strand. So eventually it'll look like the strand up here. It does this by catalyzing the formation of phosphodiester bonds between adjacent fragments. Each new double helix will consist of one old, which is the dark blue, and one new, which is the light blue, strand. This is what we call semi-conservative replication. Now let's talk about the central dogma of genetics. We've already talked about DNA replication, which is simply just propagating more DNA. But then to go from DNA, we need to go to RNA, which we do through transcription. And then we'll go to translation, which is converting RNA into proteins. So let's talk about transcription. Transcription is the process in which a particular coding region, which you can see right here, the RNA coding region, of a DNA molecule is transcribed into RNA. See the RNA transcript. To initiate transcription, several transcription factors will help RNA polymerase to bind to the DNA downstream of the title box, or the promoter region, which is right here. Many more transcription factors will bind to DNA polymerase to help it become a mature transcription complex. I'd like to go into detail about one of the transcription factors, which is the sigma factor. Sigma factor joins to the core RNA polymerase and makes the holoenzyme. Sigma factor helps the RNA polymerase find the correct promoter region to bind to. So we want to bind to here, and this sigma factor helps that. So that the transcription can begin. Once the polymerase binds to the promoter, it begins to unwind the DNA. Once unwinding is initiated, the holoenzyme leaves. The unwinding creates a transcription bubble. Here you can see the RNA polymerase, this little red circle, and sigma factor would be bound to this RNA polymerase, making the holoenzyme. And you can also see how it makes a bubble in the double-stranded DNA, so the mRNA has a chance to be transcribed. Polymerase will synthesize the complementary RNA strand, or the RNA transcript, in the 5' to 3' direction, which you see here. The newly synthesized RNA will be released into the cytoplasm once the RNA polymerase reaches the terminator sequence, which is right here. Okay, so now let's talk about translation. And remember, translation is when we go from mRNA into coding for amino acids and proteins. There are 3 main steps in translation. We have initiation, elongation, and termination. So first, the small ribosomal subunit will come and attach to the methyl G cap at the 5' end of the mRNA at the initiation site. Initiation factor 3, which is not pictured here, but it would be attached here, will stay bound to the small ribosomal subunit to prevent the large ribosomal subunit from coming to bind immediately. The tRNA will bind to the mRNA start codon, which is AUG, which you can see right here, also called the Kozak sequence. It will bind by its anticodon, which is located on the tRNA. You can see it. So you have AUG, which is the Kozak sequence, and UAC, which is the anticodon. Now elongation can begin. Initiation factor 3 will be released so the large ribosomal subunit can come bind, which will create the P site, the A site, and the E site. The first tRNA will be in the P site, and the second will be in the A site. The tRNA has the anticodon, that is the codon on the codon on the mRNA codes for. So the anticodon will be on here, which will code for an amino acid that is complementary to the codon that is located on the mRNA. Peptidyl transferase releases the amino acid from the tRNA. In the P site, the first will be methionine in eukaryotes because that's what the start codon codes for. The tRNA in the A site will be moved to the P site, and a new tRNA will be placed in the A site. So it just keeps going down the chain, like in a chain-like motion. The process will continue down the mRNA molecule until we reach a nonsense codon or a stop codon. They're the same thing, which will cause release factor, which is right here, to bind to the A site, terminating translation. The final product is a chain of amino acids that make up a protein. So we've talked about DNA replication, which is propagating more DNA, transcription, which is converting DNA into mRNA or RNA, and then translation, which is taking the RNA and making it into a protein. Thank you.